Opportunity. Such a great honor to be presenting this research from the last 2 years of my master's degree in the habitat that the plant lives in. It's so awesome. Thank you, Rachel, in specific, for the invitation to speak here. And thank you, Vera and Frank Callahan, for the brief tour of the herbarium. That was so awesome to see, and I got to see some really cool specimens of Gentneri in there as well. Like Willard said, I am a second year's master's student at OSU. I am about to defend. I defend next month. So this is kind of a little practice run for my defense. So if you have any feedback, please let me know. I think you can advance this slide, Willard.
Okay.
Thank you. Beautiful job. Okay. Show of hands, who has seen this before in person? Awesome, awesome. If you've never seen it, I think I was like snooping on iNaturalist today because I wanted to see where to go tomorrow. I haven't seen it yet today. But it's in bloom at Jacksonville Woodlands right now. So if you want to make a trip up to Jacksonville, you can see Fritillaria Gentneri in bloom. Actually, really close. They have like a little metal sculpture of it in the Brick Gardens that is kind of cool to walk by and see as well. Anyways, this is Fritillaria Gentneri. It is a perennial herb. It is a lily. It's in the Liliaceae family. It's a genus that's very closely related to Lilium. They're sister clades. It looks a little similar to that. The stems of this flower are pretty long. The flowering plants can be anywhere between 10 to 70 centimeters. It has the nodding flowers, which you'll see in several other species of this genus. It has a really striking dark red color to it. It's very beautiful. Some yellow mottling in the center. And it has a branching style. So if you look at it, the style closely, you'll notice that, and I think that that's also, you can see it a little bit in Dr. Helen Gilkey's illustration there on the left, but the style is spreading as opposed to some other species that I'm about to show you where the style is not as spreading. And you can see it around here. It's a rare endangered species. It is only located in Southern Oregon and Northern California and was only discovered officially in Northern California in 2009. Okay, you can advance the slide. Okay, so where Gentner's fritillary grows, it almost always, and I do want to say always, but I haven't, there might be some weird cases, it will grow with one of these two species. So this is Fritillaria recurva and Fritillaria affinis, and we'll go into the range a little bit more in detail later, but Fritillaria recurva grows on most of the west coast of the US, so it has a little bit more of an expansive range than Gentneri. And affinis grows all the way up on the west coast, from Canada all the way down to Southern California. So it does have a very expansive range. And of course, you can see both of these species here as well. Now that is a Gentneri. I wanted to show it very closely to a flower of recurva so you guys can see the morphological flower differences between them. Recurva has the very recurved tepals. You can't really see the styles that well here, but they're not spreading as much. And the flower shape is a little bit different. Recurva is a little bit more tubular. Affinis has the yellow mottling of Gentneri. Is pretty short when you see it out in the wild. And it has more of a cup-shaped flower to it. Yes.
Hey, Alex, which is the Fritillaria on the left?
Here, I'll you go over all the pictures. So this is Fritillaria recurva. This is scarlet lily. This is a better picture of the flower here. This is Fritillaria Gentneri, Gentner's fritillary, and this is Fritillaria affinis. Thank you for pointing that out. I didn't list the species in order. Okay, I think we can go to the next slide. So this is Louise G. Gentner. He is an entomologist, or he was an entomologist. He was born in, I think, 1890s, and he was born on a leap year. So this little newspaper article about him, when he's obviously not 20 years old, is celebrating his 20th birthday in the Oregon State College magazine. He did his bachelor's degree at OSU, and then he went over to Wisconsin for his master's degree in entomology, came back to OSU for his PhD and postdoc, and then went back to Wisconsin to teach before coming back to actually this area in Southern Oregon, in the Southern Oregon Experiment Branch Station, to do his research. He did, let's see, what is the most important thing to talk about here? So I got to go through the special collections at OSU where they have like all of his letters that he sent out. He was constantly sending bugs to other collections to identify plants, up to Dr. Helen Gilkey, the curator at OSU, who I'll talk about in a bit. He was like very passionate about his work and an amazing naturalist. He was also an incredible entomologist. I can't remember the exact species, but he released a species of beetle in this area, which basically totally killed back an invasive species of plant. He released the disease, this isn't about bugs, but he released a disease-resistant cultivar of alfalfa called Talent, that brought back the alfalfa industry to this area. He was a very talented guy. You're probably wondering why I'm talking about him right now, unless you picked up the clues from the name. He was the person that originally collected Fritillaria gentneri in order to send to Dr. Helen Gilkey to identify it. And this was him, one of the many times that he would write to Dr. Gilkey, the curator, sending plants up. This is him talking about when Gilkey is finally going to talk about him with the Fritillaria he collected. Although he actually didn't collect it originally. His daughter, Laura, collected it first in the wild. They couldn't find it afterwards. And then his other older daughter, Catherine, saw it in a floral display at a family friend's house. And after that, got to ask them and found the wild population of it. And this is what they ended up sending to Dr. Helen Gilkey. Okay, you can advance. Yes.
Do you know where they first found it?
Oh, I'm not sure. I poured through all of these special collections, and it was like they talked about Fritillaria in the collection, but not in detail, so I'm not sure. I wish I knew. I'm assuming it's probably the Jacksonville Cemetery or the Jacksonville Woodland collection. But like I said, I don't know for sure. And actually, that's a great question to lead into my next slide. So this is a picture of what I think is the collection of the plant that Gentner collected from whatever population in, I think, 48 or 49 and sent to Helen Gilkey. I only think of this because on the label, it isn't identified as a species when Helen Gilkey typed up the original label, and then later the late Ken Chambers updated the annotation to Fritillaria gentneri, but I guess it's kind of working this way on the slide. Gentner had other organisms named after him, not just plants. He had, you can see the list in the middle there, almost 5 species of bugs named after him. He was crazy about flea beetles. You can see this flea beetle on the left that was named after him. So yeah, a very, very talented entomologist. And because of his discovery of this fritillary, Helen M. Gilkey ended up naming it after the family, which I thought was very sweet. And in this 1947 letter, I think that this was even before they were talking about the Fritillaria. I think that he was just musing, oh, maybe one day we could have a plant named after us. So I thought that was really cool. Okay. All right. So we've talked a little bit about Dr. Helen M. Gilkey. She originally described Fritillaria in 1951, and we'll talk about that a little bit more later. But she was an amazing woman. I can't really say enough good things about her. She was an artist. She was a poet. She was the first woman to graduate with a PhD in botany from Berkeley, I believe. I remember it was a very prestigious school in California. And going through the special collections, I got to see so many cool things about her. Her sister Beulah, who she lived with most of her life in a bungalow style house together, after Helen passed away, wrote this like beautiful memorial about her. And she said when she walked around the herbarium, she had this air of confidence about her. And she was always like standing up straight and commanding, but also very ladylike. And she is a huge role model for me. And also in the special collections, there was a little notebook full of her poetry. And you could tell she was a truffle expert. That's what she did her thesis on. She was writing these poems to memorize the names of these truffles, the scientific names of these truffles. It was so cool. You can see age 20, one of her poems about crocuses here, and I forgot it at home. I was going to bring it, but I have a whole book of like her poems that she wrote about her gardens and crocuses and things. She was a very artistic person. She also did a lot of botanical illustrations. You can see one here. When I was going through the special collections, she had just huge sheets of things that she had traced and drawn. I know that she anonymously illustrated one of the flora of California, but she kept doing botanical illustrations until she passed away. And I wanted to show here as well in the middle a list of all the curators of the OSU herbarium and how much she went from about 20,000 specimens to over 100,000 when she was curator. And everything that I read, she was incredibly passionate about her job. So she was a really just a really awesome woman and had a lot of different skills. Okay. So, like I said, she described Fritillaria gentneri originally in 1951 off of the samples that she was given and continuously given from Dr. Gentner. And I think that it's important to mention not only in this letter to Dr. Gentner, but also in her original description in 51, she still had to clarify the differences between recurva and gentneri because they do look very similar. She said specifically that the color difference and the floral pattern is so striking, you can notice the difference between gentneri and recurva from a moving car on the highway. I also want to mention that when she wrote this, the California populations and some of the Oregon populations had not yet been discovered. And now we see populations with a lot more variation in both gentneri and recurva. This was the original drawing that she did of gentneri when she published in 1951. Okay, so let's step back a little bit and let's talk about the genus Fritillaria. As I mentioned before, the genus Fritillaria is in the lily family. They have a worldwide distribution. They're mostly in northern temperate regions, and they have like an insane amount of variability. They're really cool plants. In China, they're harvested for medicinal properties. Their bulbs are harvested for medicinal properties. And it was really cool. I just read an article about how this one plant, it basically has this like bright yellow, colorful morphotype or phenotype, and then a kind of darker one that blends into the background. And the people harvesting them are only taking the brightly colored one. So this plant is like more and more becoming this dirt color. It's really interesting. But this has led to declining populations of Fritillaria in some parts of Asia. Yeah, huge amount of variety in this genus, and you guys have probably seen there's some cultivated species as well, like just the traditional checkered lily. We have that all over our campus. I think it's very beautiful. Okay, So I'm sorry, this is so much information on one slide. My advisor doesn't like this very much, but all I want you to take out of this is these are Williams, you know them. These are all the other species of Fritillaria. These are the North American species. And their common ancestor is all the way back there. So the North American species diverged pretty early, and from what I've seen, they all tend to look, at least compared to the eastern species, a lot more similar to each other. So that's all I want you to take out of all this information, is that the North American species are very closely related to each other, more so than any other species. Yes.
Do you know, is there like a time frame-ish for that split?
I'm sure that if I actually read this paper in the entirety and then just pull the phylogeny from it, but no, I don't, I apologize. I'll have to look that up though. Okay. All right, so there's this huge diversity of species. Oregon flora recognizes 9 species of Fritillaria that are native to Oregon. I think that you guys have, well, geez, I just got to see all the specimens here. Purdyeye, Pudica, I've seen Pudica down here on a previous trip. Lauka, I know this is you, Lily. I hope it grows here. There's some really pretty species, and I'm sure that you can see them blooming in the spring. My talk is just going to focus on affinis, recurva, and gentneri. So we talked about this a little bit before, but it's really important to note that gentneri lives in the overlap of recurva and affinis. Very important to remember for later, and I just threw up the IMAT observations for the species up there as well. I think they don't let you see it on Oregon flora because it's a rare plant, but you can see it on a naturalist. And its habitat is in these woodland areas. These are some pictures that I took last year. I went collecting with my advisor. Was such a fun trip. We went up to Pilot Rock and we were just everywhere looking for these things. But within Pilot Rock, they actually kind of had a pretty variable area that they would be in. Like some of them would be closer to water, some of them would be in the sun, some of them would be in the shade. So they really didn't have this like specific niche that they would be in. In their habitat, they were kind of everywhere. So I don't know if that's important to any point that I'm trying to make, but I thought it was interesting. I know some plants just like to be in their little areas. All right. So this plant has been, there's been a little bit of argument back and forth on whether this should be a recognized species or not. I've been hinting about it a little bit, and maybe you guys already know this, but it is a putative hybrid between recurva and affinis. And I know that before the recovery plan was instated, there were some flora that weren't recognizing it as a species, either a hybrid or a subspecies of recurva, or similar. Well, now is officially recognized as a species, and in 1999 it was listed as an endangered species. I think by by Fish and Wildlife, and then someone gifted me this. This is the 2003 recovery plan in print. This is so awesome to have. I've read this back to front, but yes, in 2003, this beautiful recovery plan for Fritillaria gentneri was made, and some of the reasons for the need for a recovery plan listed were loss of habitat due to logging, agriculture, humans building houses. It was harvested for people's gardens. So there has been some population loss, and the ODA and US Fish and Wildlife have tried to start new populations and monitor them. So in 2009, actually, I think, yeah, it was in 2003. So after they published this recovery plan, the same year they discovered California populations. So they had to work in these California populations, the recovery plan, I'm assuming working with the California government on this. And then, for reasons that we'll discuss here in a second, my lab, with Dr. Aaron Liston, before I was a part of it, did some research on molecular identification techniques. So instead of looking at morphology or flower differences to identify Gentneri, taking samples, extracting DNA, and using molecular techniques to identify Gentneri, recurva, etc. And I'll talk a little bit about his prior research here in a second. But yeah, absolutely. All right, so as I said, these populations were worked into a recovery plan. And a really important part of recovering populations and making new ones is knowing how many plants you have in each population. So this is from the 2003 paper, the number of plants in each population, and trying to find if they're growing, if they're declining, if they're stable, etc. And the only point that I want to make here is that you need to count them to know how many there are, right? And to know the next year if you have less or more. Well, the problem is that, for one, sometimes these things can look a lot alike. Even just going out in the field, we would constantly see, you know, recurva with a different stem color, more similar to Gentneri, recurva with less recurved, its namesake, tepals, Gentneri that had more recurved tepals. I remember specifically me and my advisor finding what looked like a recurva otherwise with that spreading style that was only ever seen in Gentneri. Okay, so you go out to the field and these things can totally look like each other, and it makes it really confusing. The other thing is that when you go out in the field, you don't see typically these huge, like every plant is blooming, just tons of blooms of Gentneri. No, you see maybe one flowering stem, and then, I'm exaggerating a little bit, like 10 billion of these little tiny plants with 2 leaves. They are everywhere. That's because these things reproduce by their little rice grain bulblets. So do not dig one up. I'm pretty sure that's a felony. But if you dig one up, you'll see all these little tiny bulblets that'll just break off, and you can get between 50 to 100 of these little tiny individuals from just one original bulb. And they take, I mean, Frank and I were looking at his hybrid that took 7 years to bloom. These things take a long time to bloom. So even if you could use flower morphology to identify the differences between them, you would have to wait. Okay, so we have this endangered plant. We need to monitor the populations of it, but we can't do that just by going out and counting it. So what do? We use the right computer to press spacebar on. Okay, so why not use molecular techniques? This is what my advisor did in 2020, right? So Fritillaria has a secret. What you see here is genome size in base pairs. So all of the DNA that an organism has. And hopefully what you're taking out of this is the fact this is a Fritillaria affinis genome, but recurva and Gentneri have about the same genome size. Its genome is 10 times the size of a human genome. It has 10 times more genetic information than a human does. And even one of the organisms with the largest genome in the animal kingdom, the axolotl, has a smaller genome than Fritillaria. This is a pattern across the entire genus. There have been other papers published on it and talking about it, but these things have gigantic, huge genomes. It isn't rare for plants to have large genomes. I think something like 15% of plants have genomes over 10 gigabase pairs, which are huge. But this one has an exceptionally large one. Okay, so now we're going to get into the research that came before me, and then my research working on these 3 species over the last 2 years. Okay, so please bear with me here. We're getting into the weeds. A method called genome skimming was used for almost all of the analysis that you're about to see here. And I am not sure how much you know about DNA sequencing, but typically the goal for DNA sequencing is to get whatever organism you're sequencing, the entire, well, I wouldn't say typically the goal. Most of the time, if you're doing comparative genomics, this is the goal, to get the entire genome of your organism. So from chromosome 1 to chromosome 10 or whatever, and then the sex chromosomes. So you can understand important things like where genes are at, so you can compare them with other organisms that maybe have genes in the similar areas, to understand what genes are where on the chromosomes. There are a billion reasons why it's nice to have full, complete, okay, we know exactly what's in this genome. Unfortunately, you can't do that with organisms that have giant genomes. We'll go into it in a second, but it's really expensive, and it takes a lot of computational time. So everything you're about to see was done with a method called genome skimming. This is just taking a representative portion of the genome. So we have like an overall picture of what's in there. We can't tell you where things are, but like high copy number sequences, things that are in the genome a lot, we know that they're in there, and we know the proportions that are in there. I think that it's like if you took a book, instead of having a book that you could read from start to finish and you know where everything is, you have a like word cloud. So you have like all of the does, you know how many does are in the book, you know how many times the word man is in the book. You don't know where these words are, but you know they're in there. And maybe if you see the word plant a lot, you can guess that it's a book about flora. I don't know if that metaphor makes sense or anything, but the most important things are you get an idea of what the high copy number, very abundant regions of the genomes have in them, and repetitive elements, things that repeat themselves over and over again in our DNA. Okay, so it turns out this is great for chloroplast genomes. Chloroplasts have their own DNA, and we can characterize this. It's done a lot in plant science, actually, using certain loci to look for differences between individuals and between species. And Dr. Aaron Liston did this in 2022. So what you're seeing here is the RPS8 through RPS14 locus of the chloroplast genome. And on the left, you see on the top where it says R, I don't know if you can see that to the left, RAG, look at where the red is. Those are single nucleotide polymorphisms that are only in recurva. So if you sample DNA and you get DNA from this region, you can see, oh, there are those A's there instead of G's. That's a recurva. Awesome. Now we know, okay, when we take our DNA, this is a reliable way to find out, is this a recurva, is this an affinis, or is this a gentneri? Unfortunately, there was an issue with this. So this was the Grants Pass population of these 3 species. This did not carry across species. So where these single nucleotide polymorphisms are here weren't necessarily in like the Jacksonville woodland populations or the California populations. So obviously some more work had to be done on population specific polymorphisms. And like I said, this wasn't work done by me. This was work done in Dr. Aaron Liston's lab, who is my advisor. Okay, so another thing that Aaron's lab did is he did a phylogeny, which is, I guess I should have mentioned this when I put up that horrible figure, but a phylogeny is just a family tree, showing which individuals are more closely related and which ones are more distantly related. And his chloroplast phylogeny showed a really cool pattern. It showed the fact that affinis is usually more distantly related to other species, gentneri and recurva, and that populations tend to group together. So it looks like, let's say the JW population, Jacksonville woodland, that the recurva from that population and the gentneri from that population were more closely related than a gentneri from, let's say, the Grants Pass population and the Jacksonville woodland population. So you have different species in the same population more related to each other than the same species, except for affinis that was all clustering together. So we have a really interesting pattern here. And in my introduction, I said that we had 12 individuals that we sampled. That has over doubled now. Now I have 28 individuals to work with, which is really cool, from 13 populations. So what I have in the right here is all the populations that my samples are from. And some populations we were able to get all 3 species to compare, affinis, recurva, gentneri. Some populations only had one or the other. Lots of them only had a gentneri and recurva. And like I said, this is my advisor's work, the chloroplast phylogeny. This was not mine. Okay, so if we could have a beautiful, fully sequenced genome of any of these species, it would be perfect. Fully sequenced, fully assembled. We know where everything is from base pair 1 to base pair 30 billion. But unfortunately, we can't. It would be incredibly computationally expensive and actually expensive. I'll talk about that in a second. So traditional comparative genomics needs a reference genome. We need it to locate where genes are in newly sequenced species. We need it to do to find the connection between traits and genes. There are a lot of things that comparative genomics uses that we need a reference genome for. And even if we had one in the same family, we could understand where certain genes are located. But up here, finally, last year in 2024, we have a lily reference genome that they're building. This is I think it's Lilium henryi. I don't, no, it's Lilium. Yeah, it isn't henryi. I wish it was. The problem with this is it is not fully assembled. It's in like several thousand pieces. So it's about as useful as our Gentneri data. We don't have a fully assembled genome. So even though we might have all of the genomic data, we don't know where it is on any of the chromosomes. So we don't even have a starting point reference genome to compare my samples with anything in the family. I keep doing that. So used to using my laptop. Okay, like I said before, sequencing is getting cheaper, but it's still expensive. And I'm showing you kind of a misleading graph here. This is a graph for basically resequencing a genome, not initially sequencing it. But the original human genome project, which finished, I think, either in 01 or 03, cost almost $1 billion to do. And now companies in China are saying that they can sequence your genome for $100. So sequencing technology is evolving super rapidly. These are, like I said, these aren't accurate to the actual cost. My advisor just got quoted for a pine genome, and he said that it was in like the tens of thousands of dollars. So it's still insanely expensive, and that is just for raw read data. That doesn't pay for, doing the PCR and doing all the lab work, extracting the DNA. After you get the raw data, assembling it, the computational time. So these things are still really expensive, and to get a good, clean reference genome would be, especially for something with such a huge genome, would be insanely expensive and kind of out of the picture right now. Okay, we need an alternative method. We need a way to characterize these three species, see differences between them, see if Gentneri is truly a hybrid without having a full reference genome. So how do we do that? Well, one of the big pieces of this is understanding why genomes expand like this anyways. Back 100 years ago, it was assumed that we knew what DNA was and we knew what genes were, but it was assumed that all of our DNA was genes. So complex organisms like humans obviously would have more DNA because we have more genes. And really lowly organisms like lilies would have not a lot of DNA because they don't have a lot of genes. And as we started to uncover what was in DNA, C.A. Thomas, I think in the 70s, coined this term, the C value paradox, because what we were seeing is organisms like lilies with a lot of DNA were not more complex, and vice versa. We had a lot of complex organisms like us that relatively didn't have as much DNA. So there must have been something going on. And it wasn't necessarily the fact that more or less genes cause that. Now we know that genes can transcribe for different things. There's a lot of other things going on. But an important piece of this is there are things between our genes in our genome. Don't do that again, Alex. One of these things is repetitive DNA. Well, one of these things is transposable elements. Technically, genes can be repetitive as well. So what I have here is something from an awesome paper, a field guide to eukaryotic transposable elements. And you can see all of the organisms over there, humans and maize and things like that. And then those teal bubbles over at the nodes of the tree, that's the genome size. And then the circles that kind of make that half circle up there, that's the transposable element content. I'll explain this further later, but all we need to know for now is that transposable elements make up a huge part of some of these genomes. Maize, it makes up almost 75%. So think about what I said earlier. Genome skimming gives you this representation of the genome that's mostly repetitive elements. Maybe if we use these things that are repetitive and are in large copy numbers in the genome to characterize species, then we don't have to sequence everything to understand it. Another thing that can cause genome expansion is whole genome duplication, WGD. I don't want to work in that. My advisor works in strawberries and their octoploids, and he works a lot with polyploids. I'm not going into that here. The biggest polyploid I work with is a triploid. So there are other ways that genomes expand, but transposable elements are a big factor in this. So what are transposable elements? They are in almost every single eukaryotic genome to date. So we all have transposable elements in our genome. Don't freak out, they're usually not bad for you. Plants have them as well. They're pretty, at their simplest, just elements of your genome that can move around. And they typically don't do much harm. They move around, they replicate themselves. They're sometimes known as like selfish DNA or junk DNA. And by this replication technique, they can expand a genome and cause them to be very large. And what you can see here is some of the methods of transposable elements, how they move. and there's two different classes of them. There's the kind that take themselves out of the genome and put themselves somewhere else. And then there's a kind that copy and paste, replicate themselves so the original stays there and the new one moves into the genome. So another really amazing woman, Barbara McClintock, discovered transposable elements 1 year before Gentneri was published by Dr. Helen Gilkey. I thought that was a really cool connection. But she was an amazing scientist. If you have not read her book, The Feeling for an Organism, I would definitely suggest it. She has a really cool life, and she actually holds a lot of the ideas that I also hold for women in science. But she discovered TEs in the 50s by doing maze crosses and maze like linkage maps, seeing what was where in the genome. And she was constantly like, her research was pushed to the side, and she finally won the Nobel Prize for it only in the 80s. So 30 years after she originally found them, but she was such an amazing woman. I really hold a lot of respect for her. And what she worked in, like I said, was maze, and she was fascinated by these maize kernels that had this mosaic pattern. So we have maize that is dark colored. We have maize that's yellow, light colored. This is the maize that we produce and eat. But then there's this maize with two colors in the same kernel. What could be causing this? So turns out this was a transposable element. So we have a gene for kernel color. It is translated, and the protein does whatever it does. I don't know. I'm not a geneticist. Just work in genomics. But so it's normally being translated. But when a transposable element would jump into this gene and disrupt its normal translation, its normal function, the corn kernel would turn yellow. Where cells that have this gene, the TE has not jumped into yet, or it jumps out of, the gene can code for its original dark color. So we could see this in one kernel. Some cells that had the TE inserted into the gene, and some cells that did not. And that's where you get this mosaic pattern from. My advisor pointed this out. I do think it's cool. We're literally breeding corn with the TE blocking this gene. Like all of our corn is yellow. I thought that was so cool. Okay, so we can characterize these things. I think of it as like a mini ecosystem inside each one of our bodies, or every organism on Earth. Because these transposable elements, they have families, they have superfamilies, they have individuals with different protein coding regions, with different ways of excising from the genome. So when we look at an individual, each one of us has different transposable elements within us. And there have been several other studies published where they compare transposable elements across species. I think very few across individuals that I can remember, but certainly across species and genuses. And we have seen patterns where the transposable elements are different between species. So going back to Gentneri, affinis, and recurva, can we look into these things, see the different transposable elements within each one, and have a way of characterizing what is Gentneri, what is affinis, what is recurva, or do we see a pattern of hybridization with both recurva and affinis transposable elements or repetitive DNA inside Gentneri? So that's my research. We basically have just a few hypotheses for the origin of Gentneri. It was either single origin, it arose once, and there are different Gentneris in different populations. Or it has multiple origins. Gentneri has evolved in different areas. We call it the same thing, but technically it isn't the same species from one origin spreading out. It's arisen in different places, and we call it the same thing. And then, is it a hybrid or not? Okay, so these are the samples I used for analysis. We were originally doing 12, now we're doing 28, all 3 species, 13 populations. There's my advisor and I out collecting. That was so much fun. So, I know this is a lot, but basically what I want to show you here is we found lots of different transposable elements in each one of these 28 samples. So what you're seeing here is on the left, that's the original chloroplast phylogeny. So obviously, more closely related things are closer to each other. And then we have all of the species, and then we have all of the transposable elements. And I think what you can pick up here is that they all seem to be similar across all of the samples. There is no pattern between species, between populations, just at like a broad look of TE proportions, which is what this is, the proportion of each transposable element in each one of these samples. But we looked a little closer than this. So this was pretty cool. A different paper published in 2010 found, they were like looking at a bunch of different Fritillaria, and they found in Fritillaria affinis this unique satellite. I don't know if you guys have seen gel bands before, but that's what this is showing. In affinis, there is a lot of this thing, and there's not a lot in everything else. And I found that in every single one of my samples, which I thought was pretty cool. This satellite, which is a type of repetitive DNA, was in every single one of my samples. That was analyzed in this 2010 paper. That's a little aside. Obviously, there's no pattern between species or anything, but it was cool to identify this thing that hasn't been seen in a while in every single one of these guys, including Gentneri. Okay, so we still did see some patterns. I analyzed a bunch of different transposable elements and then the protein domains, so the protein coding domains within each one. And pretty consistently, I saw that FRAF was its own thing. FRAF was always off, not very closely related to everything else. Gentneri and Recurva were always pretty closely related, but I don't know. Luckily, you guys can't see those horrible bootstrapping numbers there. But there wasn't really a lot of, for sure, okay, we know this is happening in a lot of these phylogenetic trees that I did. But we looked at the rDNA and we found something really cool. So rDNA is not a transposable element. It is a gene. It is ribosomal DNA, and there is basically just a ton of copies of it in the nuclear genome. And it's been used to characterize, especially in plants, species for comparative genomics. There are some downsides to it, but I characterized it here, and it was really cool. What I was seeing was, and Dr. Stephen Myers, who was at OSU and looked at Gentneri in 2009, was seeing this exact same thing when he analyzed the rDNA. But you would get this 50-50 of these copies. So think of each one of these as an rDNA copy. So one copy of the gene, we have multiple copies of the gene within us. And at the same location, we would see different nucleotides. So that was really interesting. This is a heat map of all of my samples on the left side. And then some 10 variable locations within the rDNA that I analyzed. It's within the ITS regions. I can talk about that a little bit more. But basically, what I want you to see here is notice how recurva is mostly, where recurva is mostly yellow, which is this block right here. Affinis is mostly blue. And these guys are mostly in between. And that's literally the pattern that we were seeing, is affinis was having copies of both of these variants of rDNA within its genome. And if you look closely, you can literally see that. So this is an rDNA recurva only variant. We only saw this variant in recurva. This is an affinis only variant, that C there. And in Gentneri, we were seeing both affinis only and recurva only rDNA copies. In prior studies, this showed hybridization. I remember just reading a paper on a genus in the aster family that saw just this same exact thing. So this is like the best proof that I have of Gentneri being a hybrid that has come out of my research. Okay, and then this is kind of an aside. This was really cool. So what you're seeing here is all my samples in green here, and then closely related, this is a protein domain from a transposable element. And what you see here, closely related, the insane plate is not another lily. You can see down here, there's a bunch of other fritillarias. It's a prunus. It's apricot. And when we looked at the full, I actually found this by using BLAST and searching for the full transposon insertion, but it is almost exactly similar to the copies that are in my Fritillaria samples. So my advisor and I hypothesized that when apricot and Fritillaria shared a range, apricot picked up this insertion from Fritillaria. So this transposon, this piece of DNA, was transferred from Fritillaria to apricot, millions of years ago, which was really cool to see. Obviously that hasn't been confirmed, but I don't know, I thought it was a really cool little aside. Okay, so from our rDNA results, we see strong evidence of hybrid origin in F. Gentneri. And like I said, Stephen Myers saw this back in 2009, and I'm just confirming it. F. Gentneri constantly seems to be more related to recurva than affinis. This obviously needs more research. TEs could be a reliable method of comparative genomics. In certain cases, I found getting down to the nitty-gritty and looking at the protein domains worked a lot better than looking at the proportions of TEs, at least comparing individuals. And then there's lots more research to be done. Another master's student in our lab, M, is doing genotyping by sequencing, which should come up with some really cool results. And you guys should have them present next because they're gonna have some cool stuff. Okay. Thank you so much for having me. I didn't go over time. I was really worried about that. And this is, if you guys haven't seen it, that Fritillaria sculpture out in the Brick Gardens is so cute and so awesome. Okay, that's all I had for you. Thank you so much.
Thanks, Alex.
Yeah, of course. Do you guys normally do questions too?
Yep.
Okay, yes.
I know a bit of a story. In the 1970s, I worked at Southern Oregon Agricultural Experiment Station.
Oh,
on Hill Road. And that was where Louis Gentner spent his career as an entomologist. He had long since retired when I worked there, but he would show up pretty much every Christmas to the annual Christmas party. Almost, he was in a wheelchair. I think he was in his 90s at that time. And it was almost always brought in and accompanied by his daughter, Catherine.
Yeah.
I may have met Laura once. I can't remember her name. Never met his wife, so I don't remember her. But he obviously really cared a lot for the experiment station. The entomologist at the time, so I think Louis died in maybe the early 1980s, something like that. I'm not exactly sure, but the experiment station entomologist one day took me into his office and said, well, would you like to have Louis Gentner's basket? So I got that.
Oh, that's so
Happy
cool.
to have it.
Oh my
I
gosh.
was already collecting plants and needed places to put them before they went into the press. So I have that old fashioned basket and I still use it.
That is so cool. Wow, thanks for sharing. That's
The other
awesome.
thing I want to share is in the 1980s,
Catherine was pretty active in this chapter of the ESO. She was here most of the Thursday meetings we had right here in this room. So anyway, that's my story.
Oh, that's so cool. Yeah, I read that both the daughters, Catherine and Laura, were super active in the Jacksonville Woodlands Association in this area. And I know, unfortunately, Laura passed away 2 years ago. But yeah, they left behind a really cool history. And I know they were really passionate about the conservation of Gentneri. So it's cool that they carried on the. That's a cool story. Thank you for sharing.
Yeah.
I have
Yes, go ahead.
Chad, so my understanding is that they don't, you said they reproduce by the bullets,
Yes.
right? And so do they not make seeds, or are the seeds not viable?
So OEA did a ton of work on this back in their 09 recovery plan. And what I read from that is, yes, they got viable seeds out of all 3 species. For Gentneri, I don't know, it's a little, I don't know, I think that it seemed more inconclusive than anything else. I mean, for my purposes, Gentneri is a sterile triploid, which we see in a lot of other hybrids, but apparently they got viable seeds out of it in this recovery plan. So I suggest, if you want a definite answer on that, read the recovery plan and see, they have all the experiments in there. But yeah, they can make seeds, but they mostly reproduce with these little rice grain bulblets, because I feel like that's just works a lot better. They just break off and they go everywhere. But yeah, all 3 species apparently have had viable seeds. Yeah, go ahead.
I also met Catherine Gentner a long time ago. There is a very vague memory. So I guess it had to be in the meeting here in the early 90s. I remember her talking about bringing flowers in and putting them in the base of the living room that her mother had. And she would notice that some of them were different from others. And she said, those are the Gentneri. She picked up flowers.
You should see, I saw some old slide photos when I was doing at the special collections, and the Gentners had like these beautiful, huge groves just full of Gentneri and recurva and everything. But yeah, I'm sure they were harvesting those from native populations. But yeah, it was still, it was still really cool. And it was cool going into the history of everything. I know that Catherine died in 2007 or 2006, somewhere around there. Yeah.
All right, let's see if this.
So, before this recovery piece was written, the Plant Society gave me $200 to do a project on what were the pollinators, what were the
Oh,
visitors.
that's so cool.
And, I spent one evening in the Jacksonville graveyard. Because I had, that, powder that you can put on pollen and then see if it's been transferred. It glows in the dark as part of what makes it valuable. But I had to go in the dark to. And anyway, that was interesting.
Oh, that's so cool. So that's how you got the hummingbird photo then of the.
Well, that was actually in Jacksonville Woods.
Oh, okay. That's still cool.
Yeah, I got a hummingbird photo visiting Gentneri and then going to recurva.
Okay,
So
that's
they
so
were
cool.
visiting both. And they were saying that Andrea Beach that.
It's awesome. Is your work published online anywhere? I feel like I've read.
It's in that.
Okay, yes, all right. I've totally read that before. That's so cool. Oh, awesome. Thanks for sharing. That's so cool. Wow. Okay, we have some online.
We don't have any questions.
Okay. Oh yeah, don't leave don't.
A laser pointer, some feedback, but no questions at this point. So thank you again
Yeah, thank you.
for coming down and presenting. It was a really wonderful talk. And so one question I think Rachel had started was, what 3 books would you recommend?
So definitely go out and read The Feeling for an Organism. Yeah, the Barbara McClintock book. Oh my gosh, she had such a crazy, amazing life, and especially if you're a female scientist, like it's a handbook for me, honestly. What other two books related to Gentneri? This is kind of,
Presenters.
yeah.
These are
Oh,
presenters.
yeah, yes.
Oh, yeah.
Not question.
Yeah, I know, right?
You did suggest your colleague, your student colleague.
Oh, yeah, definitely have, I mean, M. Johnson, I have them up there. Give them 2 years, so then they can do their research first. I mean, we have so many other cool grad students doing native plant work at Oregon State University. What's her name? Abby Neat does work in fungal communities with and around Douglas fir, which is really cool. Who else? I think that is the only person that off the top of my head, I don't know if you guys have had Sebastian Mortimer come down here, the PhD student in my lab, but he's a phenomenal speaker. He does work on strawberries and he work in native plants. He works on Fritillaria chiloensis here in Oregon. So he would be another really cool presenter to have. But yeah, if you do nothing else, go out and read that Barbara McClintock book. It's so awesome.
Great.
Okay, thanks again.
Thanks again.